Hi, everyone. Here on the University of Michigan campus and those watching our live stream, welcome to Saturday Morning Physics. On the eve of the only 25-hour day of the year. I almost said 24. Okay, so enjoy that extra hour. I'm physics professor Tim Chupp, one of the organizers of Saturday Morning Physics, along with my colleague, Professor Rachel Goldman, and the team, including Carol Raybuck, and who worked so hard for us, and Monica Wood and her staff of the Warren M. Smith Demo Lab. We have a very interesting and important topic today, bringing together climate change, the Great Lakes, and human health, presented by Professor Greg Dick. But before introducing Professor Dick, I wanted to remind you that Saturday Morning Physics is made possible by you, and by your many generous contributions as Saturday Morning Physics fans. Thanks so much for helping make Saturday Morning Physics sustainable so that we can bring you important topics like today, the intersection of science and policy, like our last event on election security and last spring's discussion on the threat of nuclear weapons, cutting-edge science like our next presentation on galaxy formation, and family-friendly events featuring demos, and also who remembers the events that we had with astronaut Josh Cassada. So, Giving Tuesday, which is coming up on December 3rd, the Tuesday after Thanksgiving, actually features Saturday Morning Physics as an opportunity for your donations. And for more about supporting Saturday Morning Physics, go to SaturdayMorningPhysics.org. Saturday Morning Physics is also grateful to have the support of the Dr. Mary Lois Tiffany Endowment, the Hideko Tomizawa Endowment, the Van Loo Family Endowment, and the Pulakeshi Dayalu Astrophysics Fund. And before moving on, I want to note that today's event will include our traditional question and answer session. And for those joining us online, please send your questions to physics@umich.edu. So now I'm honored to introduce today's speaker, Gregory J. Dick, professor in the Department of Earth and Environmental Sciences and the School for Environment and Sustainability, or SEAS, at the University of Michigan. Since 2021, he has served as the director of the Cooperative Institute for Great Lakes Research, a partnership between the University of Michigan and NOAA, and Great Lakes Environmental Research Laboratory, and he's also became the director of the Great Lakes Center for Freshwaters and Human Health. As we will learn, Professor Dick's research focuses on aquatic microbiology, including deep-sea hydrothermal vents, microbial mats, and harmful cyanobacterial blooms. Did I pronounce everything right?
Yes, good job.
Thank you. Professor Dick, he grew up in the DC area, and he did his undergraduate work at the University of Virginia, and then went to California, Southern California, and received his PhD in marine biology from the Scripps Institution of Oceanography at the University of California, San Diego in 2006. After a postdoc at the University of California, Berkeley, he joined our faculty here in the Department of Earth and Environmental Sciences, and he was promoted to full professor in 2020. He's also actually a bicyclist, so I find that great, and he commutes to work by bike or by walking. So that is one thing to note, but in addition to that, he has received numerous honors and awards, including an Alfred P. Sloan Foundation Fellowship Individual Award for Outstanding Contributions to Undergraduate Education here, the Geological Society of America Geobiology and Geomicrobiology Award, and the John Dewey Award, also from the University for Teaching, and finally, also for his contributions and quality of undergraduate education, he is an Arthur F. Turnow Professor. So it's a great pleasure to welcome you back, actually, for your second Saturday morning physics presentation on climate change in the Great Lakes, the effects of algal blooms, and human health.
Okay. Thank you, Tim, for the kind introduction, and good morning, everyone. I'm delighted to be with you here today and to tell you a little bit about one of the many ways that climate change is affecting the Great Lakes, and that is how climate change is affecting these harmful algal blooms, and in turn, how those harmful algal blooms are affecting human health. So it turns out the University of Michigan has a number of fantastic organizations that tackle big problems to protect and understand the Great Lakes, and I'm really lucky to be a part of several of those, as Tim mentioned. So the Great Lakes Center for Freshwaters and Human Health, which is co-funded by the National Institutes for Health, in particular the National Institutes for Environmental Health Sciences, as well as the National Science Foundation. And then also the Cooperative Institute for Great Lakes Research, which, as Tim mentioned, is a partnership with NOAA. So a lot of what I'm going to be telling you here today is work that's being done by many people at these various organizations. So I'm looking forward to the question and answer session. Last time I gave a talk here 9 years ago in 2015, actually a question from the audience led to a whole new avenue of research in my laboratory, and probably what I would say is the most interesting paper, at least to me, that I've ever published. So no pressure, but I'm expecting some good questions that are going to lead to new research avenues. That's why I agreed to come back here and do this again. And before I really get started, I wanted to ask, how many of you have heard of these harmful algal blooms before? Maybe just a quick show of hands. Okay, so a lot of you have heard of these issues, and how many have actually experienced one? Seen it? Okay, a number of you. Anybody swam in one? Okay, so if you learn nothing else today, what I'd like you to know is do not swim in these green waters. Do not let your children swim in these green waters. And probably most of all, do not let your pets, your dogs, swim in these green waters, because it's a real health issue. And I think one of the greatest threats to freshwater ecosystems and drinking water in the world. So we're going to talk about that today. How is climate change affecting harmful algal blooms, and how in turn is that affecting human health? And I'll come back to this figure later on, but for now I just want to say that climate change is affecting these harmful algal blooms in two main ways. Number one, we have warmer waters, and warmer waters promote these harmful algal blooms. And number two, the Great Lakes region is facing more and bigger intense storm events. And these storm events are washing nutrients from the land into the lakes. And that's the other main way that climate change is affecting these harmful algal blooms. We also have good reason to think that climate change is actually affecting how people get exposed to these harmful algal blooms. So for example, we're expecting climate change to bring us higher winds, and higher winds can increase how toxins from the blooms get into the air, and how those toxins can be transported from the lake back onto the land. And so we're going to talk about how climate change is affecting the blooms and how climate change is affecting these human health exposures here today. That's where we're going. Okay, so first of all, what are these harmful algal blooms? And I'm going to use the short word for that, HABs, frequently throughout this talk. Well, they're simply overgrowths of algae, or in many cases in freshwater systems, photosynthetic bacteria. Okay, and these bacteria are called cyanobacteria, and you'll also hear them called blue-green algae. Okay, now not all cyanobacteria are harmful, and in fact, many cyanobacteria are beneficial. And in fact, last time I was here 9 years ago, I talked about how these cyanobacteria oxygenated our atmosphere on this planet. So they filled the Earth's atmosphere with oxygen, and they enabled the habitable planet that we know. They enabled life as we know it. So we have cyanobacteria to thank for a habitable Earth. But some of these cyanobacteria nowadays grow out of control, and some of them cause these harmful algal bloom problems. And so some of the problems include that there are aesthetic and taste and odor issues. They look bad, they smell bad, and they taste bad. And that alone makes these harmful algal blooms a big nuisance, right? Just look at this picture taken by my graduate student, Derek Smith, in Maumee Bay near Toledo. You wouldn't want to swim in that, hopefully, right? And again, they smell bad and they taste bad. So that alone has a number of issues in terms of recreation and tourism and people's use of the water. But the main issue of these harmful algal blooms is that they produce toxins that can be harmful and even lethal to pets and people and ecosystems. And I think one of the best descriptions of harmful algal blooms that I've read comes from one of the first scientific publications that I'm aware of about harmful algal blooms, from Francis writing in the journal Nature in 1878. He describes these algal blooms as a thick scum like green oil paint, pasty as porridge. It is swallowed by cattle when drinking and rapidly causes death. So that tells you some important things about these harmful algal blooms. One, they can be quite dangerous. If an animal the size of a cow can die from this, that tells you that this can have devastating impacts on people and pets. Number two, these harmful algal blooms are linked to agriculture, and we're going to talk about how that's the case here today. And number three, they're global. These harmful algal blooms in Australia are essentially the same ones that occur right here in the Great Lakes, the same species. And so we're going to talk about how this is a global issue. We now know, 150 years later, the exact organisms and molecules that cause these problems, okay? And in Lake Erie, and indeed in many places around the world, the main organism of concern, not all the times, but many times, is an organism called Microcystis. In particular, a species called Microcystis aeruginosa. And here's a microscope image of Microcystis. It's a little circular bacterium that's about 3 microns, 3 millionths of a meter across, that often grows in clumps that we call colonies that float to the surface of the lake and cause these, what we call scums, right? This oil paint porridge type material. And these organisms produce these small molecules, including this molecule here that's called microcystin. It's a small molecule made up of 7 amino acids that is highly toxic. And we don't know exactly why these organisms produce the toxins, but for whatever reason they produce them, and this is what causes the major health impacts. I mentioned that this organism is global, and here's a figure showing that. So here's a map of the world where all of the countries colored in green have had a microcystis bloom and toxin reported. So you see the vast majority of countries have had a bloom and the toxin reported. Those in blue have had a bloom reported and not the toxin, but I would bet you that it's just because they haven't looked for the toxin. And the ones in white, just very few countries, have not had either reported, but again, I would bet you that it's because they haven't looked for the blooms or the toxins. Okay, it just hasn't been reported yet. So I think it's safe to say that every country in the world and every state in the US faces these harmful algal blooms. And this is why about 10 years ago I decided to shift my research from deep sea oceanography to Great Lakes harmful algal bloom science, because I think this is one of the biggest threats to freshwater ecosystems and drinking water across the world. These harmful algal blooms now occur in all 5 Great Lakes. I mentioned Lake Erie, and this will be the main focus of today's talk. They occur in Lake Erie on massive scales. They occur in Lake Ontario in various locations, including this bay here. They occur in Lake Huron, especially in Saginaw Bay. They occur in aptly named Green Bay in Lake Michigan. And they occur incredibly in Lake Superior. Incredible because Lake Superior has always been what we thought of as the coldest, the cleanest, the most pristine lake, but these harmful algal blooms have been popping up even in Lake Superior in the last 10 to 20 years. So the harmful algal blooms occur in all 5 Great Lakes. The ones that we're concerned with, again, are cyanobacteria, shown in blue. There are other nuisance algae called cladophora. Here, their locations shown in green, but I'm not going to talk about that today. So let's zoom in here to the western basin of Lake Erie. Again, this is really a hot spot and what I think of as ground zero for these cyanobacterial blooms. It's ground zero because Lake Erie is the smallest and the shallowest of all the Great Lakes, and that makes it vulnerable, susceptible to these harmful algal blooms. Lake Erie is also the most socioeconomically important of all the Great Lakes, so it's the smallest and it's the most important because of the large populations of people and agriculture that are surrounding Lake Erie. On the water, it has a $1 billion fishing industry, one of the most valuable fishing industries in the world. It's a hot spot for recreation, right? Boating, fishing, beach going. And it also serves as a source of drinking water for 11 million people. A lot of people drink Lake Erie. And I'm also highlighting Lake Erie because we had this crisis 10 years ago this summer in Toledo where drinking water was compromised by these harmful algal blooms. And I want to tell you a short story about that because I think it really illustrates how vulnerable our society can be to these harmful algal blooms. Okay, so 10 years ago, this past summer, Friday, August 1st, the National Oceanic and Atmospheric Administration, right here in town, and colleagues on the East Coast, were monitoring a harmful algal bloom in western Lake Erie. Pretty typical harmful algal bloom for western Lake Erie. It had showed up about a week early, but otherwise it was unremarkable. But what happened that year is that suddenly the winds shifted and this bloom was transported from up against the Michigan coast and in Maumee Bay. So to orient you, here's the city of Toledo, here's Monroe, Michigan. Detroit would be up here to the north and the east. Winds shifted and transported this bloom to the south and the east towards the drinking water intake for the city of Toledo. So this drinking water intake is located 2.5 miles offshore from Toledo. And they have these big pipes that suck in water that then is transported that 2.5 miles to a drinking water treatment plant that serves the city of Toledo. And so the harmful algal bloom got into the drinking water system, and by 6:30 that evening, the Toledo water plant measured toxins had gotten into the drinking water system. Okay, and at 1:20 in the morning that night, this news release went out to the city of Toledo. Urgent notice to residents of Toledo and Lucas County who receive water from the city of Toledo. Do not drink the water. Do not boil the water. If you boil the water, it releases toxins from the cells and becomes more toxic. So chemists had measured toxins in the drinking water. And you can read down further here. The city advises alternative water should be used for drinking, making infant formula, making ice, brushing teeth, preparing food. I would add taking a shower. People couldn't take their medicine in the morning, right? This was a real crisis, and one of those moments when you realize how much we depend on that water coming out of the tap. And so by 6 a.m. the next morning, there was no water left in the stores of Toledo. And so people were traveling farther and farther distances from Toledo to get their water. Monroe, Michigan, Ann Arbor, Michigan, right? Walmart was out of water. This was a major crisis in a U.S.-American city that found itself without water for a long weekend. And again, it really underscored how vulnerable many of our major cities are to the quality of water in the natural environment, lakes and rivers, right? So I highlight this because the quality of our drinking water is one of the main ways in which we're vulnerable, ingesting contaminated water. Another potential exposure to these harmful algal blooms is through recreation. And so, for example, through swimming. And I mentioned, if you learn nothing else, don't go swimming in these harmful algal blooms. Here is an example of a beach in Toledo with one of these harmful algal blooms and people out in the water. And what's remarkable about this is that those people walked right by this sign that says beach closed, blue-green algae bloom present. If you look carefully, this says blue-green algae can produce toxins that can make people and animals sick. Keep out, right? So this illustrates some of the public health challenges. Even with messaging, sometimes people do not pay attention. So swimming and skin exposure is a potential route for health issues. Other types of recreational exposure, boating, for example, we now know, and this is sort of a growing awareness, that the toxins from the water can be aerosolized. I think after COVID, we're all more familiar with aerosols than we used to be, right? The idea that particles in the air can get in the air and travel and be inhaled and cause issues, right? So in this case, in the water, when waves are breaking, either caused by winds or by boating, bubbles can form and burst, and that can release particles into the atmosphere. Okay, we can then inhale those particles, and that can cause health issues. The concern here is that on a per molecule basis, the toxicity from inhalation is about 100 times higher than if you drink it. Okay, Now, luckily, there's not as much toxin usually in the air than there is in the water, so that's a plus. But still, there's a concern about what does this inhalation of toxin do to people. Furthermore, these aerosols can be transported substantial distances, up to 30 kilometers, it's thought. So now we're talking about this not just being an issue if you're on the water, but it also being an issue if you live in a coastal city, right? If you're sitting on your porch, for example, on a lake. Okay, so inhalation and boating is a concern. So we have these different modes of exposure that I've mentioned. To summarize, ingestion, inhalation, skin contact are the three main modes of exposure. I'll add for ingestion, it's not just drinking the water through a cup from the tap, but also often when you're swimming, you drink some water. And who does that mostly? I try not to drink water when I swim, but my young children, when they were little, right, would tend to drink more water than I would, and dogs. So half of all the health incidents involving people are kids 18 and under, and every year a substantial number of dogs die because of swimming in harmful algal blooms, because they don't know better, they're lapping the water up, or they're licking their fur after they get out of the water. Okay, so again, if you learn nothing else from this talk, don't let your kids or your pets in green water. So we know some of the acute effects, health effects of these toxins. Right, the day of or the next day, you're going to feel flu-like symptoms, nausea, vomiting, diarrhea, fever. There can be neurological symptoms and headaches. It can irritate your skin, and it can also cause respiratory issues. We know far less about chronic effects, right? So what if you're breathing this stuff all summer for your whole life? Right? What if you live in a coastal environment? We know far less about that because it's much harder to study. But there is some epidemiological evidence that it may be linked to neurological issues like ALS. There is laboratory evidence that these toxins are carcinogens. They can increase the chance of cancer. It can affect your brain, and also good evidence that it can cause respiratory issues. It can exacerbate asthma, and people with asthma are more vulnerable to the health effects of these toxins. Okay, so we've talked about what these harmful algal blooms are. We've talked about some of the health concerns. What causes these harmful algal blooms? Why are they there? Well, a main cause of these harmful algal blooms are nutrients. We don't tend to think of nutrients as pollution, but here's a case where an excess of nutrients is allowing these algae to grow out of control. In the 1970s, when we were debating, not we because I was a baby, but the scientific community was debating what nutrients are causing these harmful algal blooms. Is it phosphorus? Is it nitrogen? Is it carbon? The main way that they nailed this down is by doing whole lake experiments. So Canada has this fantastic experimental lake area where they've dedicated, Canada has a lot of lakes, so they set aside a few and said, we're going to dedicate these lakes to doing scientific experiments. And this is really important because ecosystems work in a different way than laboratory bottles work, right? So by doing experiments on whole lakes, we can understand the effects of different environmental conditions on the whole ecosystem. And so here's an example, Lake 226, where they divided up the lake. To one half of the lake, they added phosphorus, nitrogen, and carbon. To the other half of the lake, they added only nitrogen and carbon. And so by repeating this experiment in many places and in many instances, they found that only when phosphorus was added did it cause these harmful algal blooms. So really strong evidence that phosphorus is a primary driver of these harmful algal blooms. Okay, so where does the phosphorus come from, right? Historically, the phosphorus came from detergents in laundry detergents, phosphorus in laundry detergents, and human waste from wastewater. And we solved those problems in the 1970s, but now most of the phosphorus comes from agriculture. Okay? And I'm going to show a quick video clip that illustrates where this phosphorus comes from. This is from the terrific PBS show Great Lakes Now. And also it's an excerpt from David Ruck's wonderful documentary called The Erie Situation. So if you're interested in learning more about this, I highly encourage you to check out this episode and this documentary. So let's have a listen here to this show.
Maumee River watershed drains 5,000 square miles within Ohio, 80% of which is used for agriculture. Phosphorus is a key mineral needed for crop growth, and lots of it is applied to fields in the form of fertilizer and manure. But when rain washes it off of fields and into the river, and the river delivers it to Lake Erie, phosphorus can boost the growth of cyanobacteria and fuel algae blooms.
What we know from the Ohio EPA and other data is that more than 90% of the phosphorus that gets into western Lake Erie comes from agricultural runoff. And that's really from two places. That's CAFOs, that's concentrated animal feedlot operations with thousands and thousands of chickens and pigs and cattle. And then that's fertilizer that's spread on the corn and soy fields. Rainstorm comes, it runs off, it gets into the tributaries, which go into the Maumee River, and then make their way into western Lake Erie.
Okay, so that's Howard Lerner, who's at the Environmental Law and Policy Center, one of the terrific environmental organizations that's working on this issue. So let's take a closer look at where this phosphorus is coming from. Here's some data. It's a little bit dated, but it hasn't changed all that much. So this is showing where the phosphorus that goes into western Lake Erie is coming from. And the blue arrows represent the tributaries, the rivers that are delivering phosphorus into Lake Erie. And the size of the arrow represents the amount of phosphorus, and that amount of phosphorus is also shown in a number here. So this is 2,600 tons of phosphorus that's coming in from the Maumee River, the biggest arrow, right? And what's remarkable about that is the Maumee is a big river, but it's not nearly as big, for example, as the Detroit River. The Detroit River has 20 times more water, but the Maumee River is delivering most of the phosphorus because of all the intensive agriculture that takes place in the Maumee River watershed. Okay? And then if we look at where that phosphorus is coming from on land, almost 90% of it is from non-point source pollution. That means it's not coming out of some pipe at a wastewater treatment plant. It's coming from non-point sources, meaning diffuse sources like farms, okay? Big areas. And when you break that down into the different sources, the total phosphorus at least, about 25, this number's gone down a bit now, but around a quarter is coming from wastewater treatment and sewage. 25% of it is coming from the waste of animals, and about 40% of it is coming from fertilizer that's put on land, okay? So the vast majority of this is coming from agriculture. And another remarkable stat here is that 1 ton of this phosphorus, because phosphorus is just a small part of the cell, that translates into about 500 tons of bacteria. So we're talking about a lot of algae that can be produced off of this phosphorus. So phosphorus is a major driver of these harmful algal blooms, but recent science also indicates that nitrogen also plays a role. So another nutrient that's used as a fertilizer. And this is work that's done by my colleagues in the Great Lakes Center for Freshwaters and Human Health, especially Hans Pearl and Justin Chafin and their colleagues and students. And they do some of these experiments. Lake Erie's too big to experiment on, so we have to scale those experiments down, but the Ohio State University has this fantastic facility where they have these big tanks that they can do tank-size experiments on. So they can, in one case, leave the tank alone, and we call that a control. In other cases, they can add just nitrogen or just phosphorus or nitrogen and phosphorus. Okay, so here's a case where they've added nitrogen and phosphorus, and you can see that that's where we get the biggest response. Okay, so this is providing evidence that the algae are what we call co-limited by both nitrogen and phosphorus. So both nitrogen and phosphorus are in limited supply. They're in short supply. Okay? And this and other science like it has been used to come to the conclusion that we really need to think about managing both nitrogen and phosphorus. And as Hans likes to say, it takes two to tango, right? So phosphorus alone results in a certain response. Nitrogen alone results in a certain response. But an excess of nitrogen and phosphorus is really where we get the biggest response. And so we need to think about limiting both of those nutrients. So it's the amount of nutrients that we're putting on fields and that are coming out of animals from agriculture, but it's also how we have changed the landscape in this part of the world. And so here I want to emphasize that we've transformed this part of the country, or at least northwestern Ohio, from what used to be a giant black swamp, what was called the Great Black Swamp, into farm fields, right? So this used to be a swamp that was about 40 miles wide and 120 miles long, about the size of a small state like Connecticut, right? And we drained that swamp, we cut down the trees, and we converted it into some of the most fertile farmland in the country and in the world. And in doing so, we removed one of nature's best way of removing nutrients from water, right? These swamps slow down the water, and the vegetation takes up the nutrients. It removes the nutrients from the water. And so in converting a swamp into farmland, we have replaced what scientists like to refer to Lake Erie's as Lake Erie's kidney, the thing that removes the pollutants. We've replaced that with farms and then this series of drains and ditches that funnel water directly from the farm fields to Great Lakes and rivers, fueling these harmful algal blooms. So Lake Erie is a great example of how changes in land use and conversion of natural systems to agricultural systems has had major impacts on our natural waters. Okay, so let's turn our attention now to Lake Superior, because Lake Superior is interesting in that these harmful algal blooms are popping up without big changes in land use. Okay, Lake Superior we've always thought of as this cold, clear, low nutrient, pristine lake. And we haven't seen big changes in land use in the Lake Superior watershed, and yet these harmful algal blooms have started popping up. So what is going on there, and what can we learn from this? This is a question that my colleague Bob Sterner at the University of Minnesota has been studying, and he published this paper in 2020 presenting evidence that the harmful algal blooms in Lake Superior are driven by climate change. And so let me walk you through this figure here. This figure shows the cumulative number of days where the surface waters of Lake Erie have gotten above 10 degrees C. That's not that warm, but it's warm for Lake Erie, about 50 degrees Fahrenheit. And so this is showing from June, July, August, and September, the cumulative number of days that are above 50 degrees Fahrenheit, 10 degrees C, in Lake Superior. And what you see here, labeled in red and blue, are those years that had major blooms or minor blooms. And so the big blooms tend to occur in the warmest years. So this is indicating that blooms occur in warmer years, and this is linking temperature to algal blooms. The other way that this is linked to climate is that the two biggest bloom years, the major blooms, 2012 and 2018, those blooms were preceded by major rainfall events. These were historic storms that caused historic flooding and damage, but they also are thought to have washed heavy loads of nutrients from land into the lake. Okay? So major rainfall events tend to influence the bloom severity. Okay? So this combination, this double whammy of temperature and rainfall seems to be driving these harmful algal blooms. And it turns out that Lake Superior is among the fastest warming lakes in the world. So here's a study in 2015 that quantified how fast lakes are warming up. And you can see that around the world, lakes are getting warmer, good for swimming, but bad for harmful algal blooms. So bad for swimming in that sense. And Lake Superior is a real outlier. So here are the eastern and central basins of Lake Superior. They're warming at about double, or in some cases triple the average rate. And that's also true of many of the other Great Lakes. They're warming at a pretty rapid rate, about a degree per decade. Okay, so it doesn't sound like a lot, but for reasons I'm going to show on the next slide, this makes a big difference in the ecology of the lakes. Okay, so why do these harmful algal blooms respond so sensitively to changes in temperature? Well, it has to do with the physiology of cyanobacteria. So here on the left, I'm just showing like Lake Superior. Many of the other Great Lakes are also getting warmer. And on the right, I'm showing the physiological characteristics of cyanobacteria and their competing algae that explain why cyanobacteria are taking over in a warming world. Okay? So what we're looking at here is how fast these different microorganisms grow in response to temperature. Okay, so temperatures on the horizontal axis and growth rates, this is as a percent of their maximum growth rates, are on the vertical axis. And what I'd like to draw your attention to is that cyanobacteria have a much higher growth optimum in terms of temperature than their competing algae. Okay, so these other algae are their competitors, and these are what we think of as the good algae, the ones that transfer food up the food web and don't cause problems in terms of algal blooms and toxins. So consider what a 2-degree change, if we look at Lake Erie's gone up about 2 degrees over this 20 to 40 year period here. So let's just say from 1980 to 2020, Lake Erie has gone up about 2 degrees. So we've shifted from here to here on average in Lake Erie. And so that is at a higher, at only 2 degrees is quite a big difference in cyanobacterial growth rate. These bacteria are growing much faster at higher temperatures. Now contrast that to the other algae that are competing with the cyanobacteria. These dinoflagellates, right, we increase by 2 degrees, that actually reduces their growth rate. The diatoms, one of the big competitors for cyanobacteria, 2-degree difference, that reduces their growth rate. It inhibits their growth. And so what we're seeing is with increasing temperature is a change in the competitive landscape between cyanobacteria and the so-called good algae. Okay? And this is allowing cyanobacteria to take over and to grow, to win that competition. All right, so that's an example from Lake Superior and Lake Erie. And then a recent paper just from June of this year quantified this effect in western Lake Erie. And it showed that due to this increase in temperature, cyanobacterial growth rate has increased by up to 30% over the last 20 to 30 years or so. And it's not just how fast they're growing, but it's also the length of time during which they're growing. These cyanobacteria tend to grow fastest once it hits about 17 degrees C. And the window of time in which Lake Erie is 17 degrees C has been getting longer. And this is familiar to all of you. The leaves are coming out earlier, right? The flowers are coming up earlier. The leaves are staying on longer in the fall, as we saw this year. Well, same thing with the cyanobacteria. Their growing season is getting longer. And this study quantified that bloom season has expanded by up to a month over the past few decades. Okay, so we can see it in the data. We can also see it in the headlines. So here's a headline from the Toledo Blade from 2022. A long, strange bloom. Lake Erie algae acted very unusual this year. This was published in November because in 2022, the algal bloom went into November. Almost unheard of. Usually they peter out in late September or early October. This year, harmful algal blooms are getting worse because, this was in June, this year set the record for the earliest harmful algal bloom in Lake Erie on record, June 24th. And also this year, a headline in mid-October that the algal bloom is still 80 square miles in mid-October. And it was present still about a week ago. I haven't checked yesterday to see if it was still here into November. But chances are we still had this algal bloom in November. So again, blooms are getting longer. Okay, so that's temperature. Let's now consider the other main climate effect that I mentioned, which are extreme precipitation events. And again, you can see this in the headlines, and I probably don't have to tell you that we're experiencing more flooding in this warmer world, right? Last year, there were these floods in Metro Detroit, right? The airport was shut down. This article was about how floods are a recurring problem in Metro Detroit. People's basements are getting affected. Roadways are getting affected. So this is a big problem in this part of the country. Also think about what happened in Asheville, what's happened in Spain, right? You're seeing these events play out across the country and across the world. And so here's some statistics from 1958 to 2016, the observed change in extreme precipitation. This is the amount, this is the events, the heaviest 1% of events, okay? So the most extreme precipitation events. They're increasing across the country, but especially in the upper Midwest and the Northeast. There's been a 42% increase in extreme precipitation events in the Great Lakes region. Here's what that looks like on a finer time scale. So again, just showing that the number of these extreme precipitation events has really increased as we've gotten into the 1990s and the 2000s. So how does this affect harmful algal blooms? Well, something like 80% of all the nutrients that get into the lake are washed in during the 10 largest storm events each year. So these big storms have a disproportionate impact on the nutrient loading to the lake. And again, it's these nutrients that are causing the harmful algal blooms. We have a really good example of this. In 2011, we had a record rainfall event that caused the biggest cyanobacterial bloom ever recorded in Lake Erie. It was a 10 out of 10 until 2015 when we had an even bigger bloom. And the concern moving forward is that the increasing frequency and intensity of these extreme precipitations is expected to continue to rise. Okay, so for example, by mid-century, we expect 1-inch precipitation events to increase by 20% and 2-inch precipitation events to increase by about 40%. Okay, so this double whammy of temperature and precipitation is expected to continue. Okay, so that's how climate is affecting harmful algal blooms. Higher temperatures and more precipitation means more nutrients and bigger harmful algal blooms. It means a longer bloom season. It means more intense harmful algal blooms. And one thing I only briefly mentioned is we also expect with climate change increasing wind speeds. Okay, increasing wind speeds could mean more aerosolization, right? Causes more waves, which causes more of those toxins to get up into the air. It could also affect how the toxins are transported through the air. Okay? So all of this, longer blooms, more intense blooms, more aerosolization, could mean more toxin exposure, is what we're concerned about. The other thing that we're concerned about are that there are emerging toxins. So it's not just microcystin that's out there. There are other toxins that we're concerned about, including saxitoxin, which we just found evidence for is linked to higher temperature. So I'd like to tell you a little short story from my laboratory about how these emerging toxins might be an issue going forward. So this is a slide, and I'm going to present work from my PhD student Lauren Hart, who is an expert at identifying these chemical compounds that are in natural ecosystems. And her data and others' recent data show that in fact these harmful algal blooms are producing a wide variety of different molecules. It's not just the microcystins, but they're producing other compounds, in some cases other toxins. And in some cases we know these toxins, we've discovered them before, but there's also evidence that there are a bunch of unknown compounds that we have yet to discover. So we're still discovering these molecules. In some cases they're concerns. In some cases there are opportunities here. So we're looking to discover new antibiotics and new anti-cancer compounds from this chemical diversity. But we also need to worry about tracking these emerging toxins. And one of the main ways that my laboratory does that is with environmental genomics. Okay, so let me tell you briefly about what we're doing in the area of environmental genomics. This allows us to track which organisms are there and also what toxins they're capable of producing. Okay, so let me tell you a little bit about how this works. We would take a sample of a harmful algal bloom. We would extract the DNA directly from that water. We would randomly sequence that DNA, and DNA sequencing technologies these days are amazing. We can produce billions and billions of DNA sequences, okay? Much cheaper than we used to be able to do it. And we can take those DNA sequences, and this is like the world's biggest puzzle, we can piece them together based on shared DNA sequences, the A's and T's and G's and C's that compose DNA. And when we do this, and we use some other methods, we can actually assemble those DNA pieces into whole genomes. So we have now the whole genome sequence of the microorganisms that are causing harmful algal blooms, even though we never had to culture those organisms. We never had to grow them in a laboratory. We've just sampled them and sequenced their DNA. And this is exciting because it allows us to track which organisms are present in that harmful algal bloom. What do they need to live? What are their nutrient and energy requirements? And what toxins are they capable of producing? Okay? So this is something that Lauren has really focused on. What toxins can they produce? And this is a big plot, but I'll orient you to it. And what I want you to get out of it is that this is showing the occurrence of the genes that encode 9 different cyanobacterial toxins and related compounds. Okay? 9 different compounds. And so one of these here is microcystin, but there's 8 others as well. And it's showing their abundance at 4 different locations in Western Lake Erie. These are our sampling stations, WE12, 2, 4, and 8, at different sampling dates. So we're out there every week during the bloom season. And the bubbles are showing the abundance of the genes, the size of the bubbles. And this is from 2016 to 2020 is the data, okay? And the main thing that I want to communicate here is that these 9 different toxins, at least their genes, are co-occurring at many different times. And so this is indicating that multiple cyanobacterial toxins and products are likely present. This is a concern because there's evidence in the literature that mixtures of these toxins can actually be more toxic than any one toxin or even than the sum of the toxicities of the individual toxins. So there are synergisms to the toxicity. If you get hit by multiple toxins, it's worse than getting hit by one or the other or even both combined. And then I want to draw your attention to one particular toxin called saxitoxin. Saxitoxin is a neurotoxin. It affects the brain and the nervous system. And it's famous, you might have heard of paralytic shellfish poisoning, right? If you eat contaminated oysters, you could get paralyzed and it can even cause death. So this is a really potent toxin. It's so potent that it's actually registered as a chemical warfare agent. It's one of the most potent natural toxins known. And we're seeing the genes for it in western Lake Erie. Now, we're not seeing a lot of the toxin, so it's not at high enough levels to cause major health issues, we don't think. But the occurrence of the gene is a concern because historically this has not been present in Lake Erie. And so my graduate student, Paul Denial, went deeper into this genetic data, and all these arrows here represent the genes that encode the biosynthesis, the making of that saxitoxin. Okay, so these are think of these as like molecular markers of the presence of that toxin. So we know it's there, and Paul's work was the first to identify that indeed all of the genes that you need to make saxitoxin are present in Lake Erie. With this data, he was able to identify which organism is producing the saxitoxin. It's not Microcystis, it's a different cyanobacterium called Dolichospermum. They don't make these easy to pronounce. And then the other thing that Paul was able to do is he looked at where are these genes occurring and what environmental factors is the abundance of these genes linked to. And one of the two strongest correlations was that the abundance of these saxitoxin genes correlates with temperature. So at higher temperature, you get more saxitoxin genes. So the concern here is saxitoxin going to become more abundant as Great Lakes waters continue to warm up. So this is what we would refer to as an emerging toxin, right? Hasn't historically been in the Great Lakes, but with warming waters, it seems to be coming in and maybe getting more abundant, okay? So that's another effect of climate change that we're concerned about. Okay, in the last 5 to 10 minutes here, I want to deal with this question of what do we do about these harmful algal blooms? How can we deal with harmful algal blooms? And the first, I'll talk about 3 main strategies. The first is to improve our scientific understanding of harmful algal blooms, because with better understanding of these harmful algal blooms, we can come up with policies that address the harmful algal blooms and also actions that can mitigate harmful algal blooms, like developing better health treatments and better ways of treating health issues associated with harmful algal blooms. And this is one of the main goals of our renewed Great Lakes Center for Freshwaters and Human Health at the University of Michigan. So this is bringing together lake scientists and microbiologists and also biomedical scientists to ask, how is climate change going to affect the growth of these harmful algal blooms? How is it going to affect, for example, the changing winds? How is that going to affect how these toxins get up into the air? How they're transported from the lake onto the land? And finally, how those toxins affect people's health. Okay, so really exciting that we're putting together the environmental scientists with the biomedical scientists. This project also has a community engagement team where we're looking at how can HAB's research and data and information be used most effectively to support and protect vulnerable communities. And I'll tell you some examples of that here in just a moment. So this involves 11 different universities. We've got University of Michigan and Ohio State and Michigan State University all working together in terms of collaborations, and also with the federal agency NOAA Great Lakes Environmental Research Laboratory. And again, this is jointly funded by the National Institutes for Health and the National Science Foundation. So I'll give you just a little bit of a sense of the type of work that we're doing here. So first of all, this new center is creating the first ever harmful algal bloom aerosol sensor network in this part of the world. So what we're doing here is trying to address this question of how much toxin is getting up into the air. Okay, we're collaborating with the Lake Erie Center and Tom Bridgman and other colleagues at the University of Toledo, and we have this new Lake Erie high volume air sampler station. So basically it's sucking in air and filtering the air and capturing toxins that get onto that filter. We then take those toxins over to another laboratory at the University of Toledo, where my colleague Dragan Isailovic does mass spectrometry to identify the toxin compounds. Okay, so now we can quantify how much toxin is in the air, and we can use that information to say, okay, what are people being exposed to? Each of us breathes in thousands of liters of air per day, right? So we can calculate how much toxin we're being exposed to, and that can be paired with experiments in the laboratory to look at how much toxin does it take to make lung cells respond, right? How much toxin does it take to have health impacts, and what are those health impacts? So here we're working with the University of Toledo Medical School. So here's some pretty pictures of that air sampler. You can see that it's relatively small, so it can be moved around. It's mobile, so this can be deployed to different locations to assess how is climate change affecting the transport of toxins through the air. Another important component of our new center is that it has a community health study to help provide evidence-based medicine guidelines for harmful algal blooms. So here we're testing the hypothesis that exposure to these harmful algal bloom aerosols adversely affects the lungs of people that play and live and work near the shore. Okay? So this is enrolling about 200 patients from the counties of Lucas County, Ottawa County, and Sandusky County in Ohio. It's a 5-year study where people that are exposed are going to be sampled and tracked to understand what type of exposure are you experiencing and how is that impacting your health. That research is also being translated to public health policy. So here my colleagues, I should have mentioned this is David Kennedy and Stephen Holler and their colleagues and students at the University of Toledo are also really engaging with the medical community, okay? So not just researchers, but also practitioners. So they're engaged with the Ohio State Medical Association. They've developed new policies, this policy for increasing awareness of harmful algal bloom toxicity, where they're interacting with medical doctors and medical students to raise awareness about these health issues and raise awareness about identifying and treating these health issues. Okay, so those are some examples of the types of science that we're doing to understand and address harmful algal blooms. Another approach that we're taking is to provide early warning systems to stakeholders. So here we're monitoring and also forecasting these harmful algal blooms. So here's where the Cooperative Institute for Great Lakes Research and the NOAA Great Lakes Environmental Research Laboratory are out there, and other organizations, but here I'll highlight Sigler and Glurl, are out there on the water collecting samples and analyzing the dynamics of what's going on out there in the lakes. So here, for example, is my student Lauren Hart out on the water collecting samples. We have a whole team of staff at the Cooperative Institute for Great Lakes Research and at Glurl that are out there every week from April until October, in some cases November, tracking what's happening out there in the lake. Now there are limitations to what humans can do, right? We can't be out there 24 hours every day. And so we're also developing autonomous sampling and monitoring platforms. So these are robots that don't have people, and in some cases don't even have people controlling them, but they're autonomous. They're out there swimming around the lake or sitting in place, collecting samples around the clock and transmitting that data back to shore. So this is environmental sample processors, gliders and autonomous vehicles, and also buoys that are out there in the lake collecting data in terms of observing systems. On the human side, just to give you a sense, we just wrapped up the field season a couple weeks ago, so I don't have the numbers for this year, but last year, this effort represented about 51 boat cruises, 16,000 total samples taken. To process those samples was about 600 hours of filtering water, and that was fortunately spread across 22 different people. But they measured 30 different water quality parameters, including the amount of toxin that's in the water. And many of those parameters, including the amount of toxin, is made publicly available within 48 hours. So this is really important work that one of our federal agencies and their academic partners are doing to get this data out to the public. And in particular, we have a fantastic research engagement team. So these are professionals that take that data and work with stakeholders to make sure that the data is getting into their hands and being used most efficiently. Okay, so research and monitoring at Sigler and Glurl helps stakeholders make informed decisions about harmful algal blooms. So we call these decision support tools. So these would be things like forecasts. So like the weather forecast, right? We have harmful algal bloom forecast, the HAB tracker, which shows you where the bloom is now and where it's expected to go in 5 days. Okay, this is using computer simulations looking at currents and winds and so on. And this information is transmitted and shared with people like drinking water intake managers, with governments, with fisheries and ecosystem managers, with charter boat captains who are out there taking clients out in the water, need to know, where do I go to avoid the harmful algal blooms? We share this information with policy and decision makers, all to help them make decisions, like is it safe to swim here? Is it safe to kayak here? Should we adjust the way we're treating the water for drinking water? What policies should we be implementing, and how might algal blooms be affecting the food web and fisheries? Okay, so this is a really good example of how the University of Michigan and their partners are doing science for society, not just for research purposes. Okay, so that's how we can understand the blooms and how we can provide warning, right, to people. But really what we want to do at the end of the day is prevent these harmful algal blooms. So how do we prevent these harmful algal blooms from happening? Well, most of the science, I should at least say, we have a good understanding of what we need to do. We need to reduce the amount of nutrients that are going into the lakes. And unfortunately, U.S. environmental laws, in particular the Clean Water Act and the Great Lakes Water Quality Agreement, are not sufficient to address this non-point source pollution, or in this case, nutrients from agriculture. They were great for addressing point source pollution, like phosphorus from detergents and phosphorus from wastewater treatment plants, but they haven't been successful. They're really not set up to address non-point source pollution. So the main way that our government has been trying to address this issue is by incentivizing farmers to use best management practices. These are practices like applying the fertilizer below the soil instead of on top of the soil. Having a buffer between your farm and the stream, right? Installing wetlands to deal with the water coming off of your farm. And at least nationwide, a long track record shows that these incentivization procedures have not worked. In fact, our government has spent probably by now over $30 billion on voluntary incentive-based programs since 2009. And that has made a large number of farming operations more environmentally friendly. The problem is that at any given time, it's a relatively small fraction of farms that are engaged in these best practices. According to this article on the Associated Press, only about 6% of the 2 million farms nationwide are enrolled in these incentive programs. Now, there is a program right now in Ohio. Ohio is investing upwards of $100 million per year in these best management practices in a program called H2 Ohio by the governor there. And there is some evidence that that is working, but we're still unlikely to meet targets we have for reducing phosphorus by 40% by 2025. So it's not working fast enough, although I do hope that we get there. And what I want to point out here is that this is a case where regulators are seeking voluntary cooperation instead of mandatory limits on nutrients and how they're getting into the water. So it's a real contrast to most industries. We don't usually allow industries to voluntarily reduce pollution, right? And of course here it's because this is for growing food and this is farming, not something that we usually think of as pollution, but in fact it is. Okay, and then I want to wrap up with a couple of thoughts about what we can all do to limit nutrient pollution. And the first thing here is your diet. Watch what you eat. And why do I say this? Meat requires at least 10 times more water, fertilizer, and land than plants do on a per calorie basis. And I don't have any delusions that I'm going to convert a lot of people here into vegetarianism, but we don't need everybody to be vegetarian. If we would all just eat about half as much meat, we'd get where we need to be, right? Growing a lot less food, using a lot less fertilizer. So we know that industrial corn production is the major source of nutrient pollution. About 36% of that corn that we grow isn't to feed people, it's to make animals. Another 40% of the corn that we grow nationwide goes to make ethanol that we put in our gas tanks, right? You go to the gas station and you see that ethanol blend. Ethanol was added to gas for good intentions. One, to get our nation energy independent, and two, with the thought that biofuels are better than fossil fuels. But now the US produces more oil than any other country, so we're essentially energy independent. And we now know that we'd be better off drilling for oil than producing this ethanol from a carbon emissions perspective. Okay, so it's not good for the environment. We don't really need it. It's a bad policy. And it's causing these water issues that we've talked about here today. So what can we do about that? Well, one of the things that I would say is we should support advocacy groups like the Healing Our Waters Coalition, the National Wildlife Federation, the Nature Conservancy, the Environmental Law and Policy Center, the Alliance for the Great Lakes. These are fantastic organizations that educate and promote environmental policies that protect our waters. Okay? So these organizations are responsible for a lot of the funding that comes in to address these issues, both to do the research and to directly address the issues. And then finally, I thought it was fitting to, with Election Day coming up, to say we can all vote as well, right? Fortunately, the Great Lakes are a bipartisan issue, one of the few bipartisan issues that are left, and that's because people on both sides of the aisle love the Great Lakes, and so our politicians on both sides of the aisle also love and support the Great Lakes. But that being said, there are certainly big differences in to what extent our politicians support water and environmental policies, right? And an organization that does a great job of tracking all that is the League of Conservation Voters. You can go on that website and see how different politicians have done in terms of their environmental records. Okay, and with that, I'm going to stop because I'm out of time, and thank you very much for your attention. And of course, I'd be happy to answer questions.
Oh, well, thank you so much, Professor Dick. And we will go to the question and answer period. You know, you reminded me of the movie Jaws, in a sense, not because algae look like sharks necessarily, but because of the theme of that movie, if people are familiar with it, that there is this tension between tourism and using the environment and the danger of it. So that was great. So we'll just take a few minutes to get set up here for the question and answer. Those online, remember that you can send, and in fact those here in the room, if you'd like, can send your questions to physics@umich.edu, and we will receive them. Ready? Okay, so let's start over here.
That was a great talk. Do these algae blooms ever happen in rivers? And is the reason why because they run too fast, or is there another reason?
Yeah, exactly. So the question was, do these algae blooms ever happen in rivers? And the answer is sometimes, but only when the water slows down enough that the algae don't get washed out. So flowing water is not conducive to algal growth because it's moving too much, and because it's called the residence time, how long that water is sitting there, is too short. So the algae just don't have enough time to grow as that water is moving along. So where you'll see algae blooms occurring is in like when the flow slows down, or in impoundments, like where you have a dam and the water movement is reduced. Yep, good question.
I'm very much concerned about the accumulation of these toxins in the lake. You're talking about maybe a billion pounds of algae each year. What happens to the toxins? Do they accumulate over time?
Yeah, that's a great question. And a couple of years ago, we did what we called a HABS grab, where all of our institutions and colleagues went out on the same day and sampled the lake to try and cover it spatially at the same time and measure toxins to quantify how much toxin was in the lake that day. And you're right, it's an amazing amount of toxin. I forget the number, but it was like a ton of this toxin. And so what happens to that toxin? Where does it go? And there's been quite a bit of research lately on this, and the answer is that the toxin is degraded by bacteria. So it is degraded on relatively short timescales, on the order of days. So as soon as that toxin is no longer produced, it is going to be chewed up and eaten by other bacteria that are literally using that toxin as a source of energy and nutrients. And there's a lot of interesting research going on into can we harness those bacteria to treat drinking water. So there's some technologies that could come out of these toxin-eating bacteria. Yeah, good question.
Follow up to the last question here. You had shown a slide that had the tons, I believe, of phosphorus coming into Lake Erie from various watershed sources.
Yep.
And it appeared, if I saw it right, that there was a similar amount coming from the Maumee River as there was from the Detroit River. And I know as a frequent boater in that area that we're fortunate that we rarely have algae blooms in the mouth of the Detroit River area. And I think from what the last response was, is that due to mostly the flow there, that has a greater flow, or is it temperature related, or some other? Yeah, there it is.
Great question.
2617 versus 22.
Great question. Yeah, so the observation is that, we've got almost the same amount of nutrients coming from the Detroit River and from the Maumee River. Why don't we have more blooms up here, right? And the answer is in part because of the flow. So if you've tried boating up the Detroit River, you'll see that that's some pretty good flow coming through there, right? So the water's moving. But the other issue is, as I mentioned, this is more than half of the nutrients in the Maumee River, and yet there's 20 times more water coming out of the Detroit River. And what that means is that the nutrient concentrations from the Detroit River are much lower. And so it's not just the amount of nutrient that matters, it's the concentration of nutrient, because of ecological and biochemical reasons that they need a certain concentration to efficiently bring that nutrient into the cell. So I'd say it's both. It's the flow rate as well as the concentration. And there's some debate about whether the phosphorus coming out of the Detroit River is even causing harmful algal, is contributing to the problem or not, because of those low concentrations. Once that water gets into the basin, it is slowing down, so that phosphorus could be available for algal uptake, but the concentrations are so low that most of us think that it's primarily this stuff coming out of the Maumee River that's causing the HABs. Yep.
Is some sort of mixing strategy viable on that scale, like a big fan in the river?
That is a great question. So my colleague Tom Bridgman, director of the Lake Erie Center, has proposed that we build a big wall in Lake Erie here, because usually what happens is the Detroit River water kind of takes a, I guess it would be a left turn, and comes out this way through the islands north of the islands, whereas the Maumee River water comes in here and stays in the Maumee Bay and exits the islands down here. So he has proposed that the Army Corps of Engineers should build a wall here to divert the water down and basically flush out the basin. So that would be an engineering approach to fix the problem. He's serious about it, but most people think, once we start tinkering with things, who knows what's going to happen. It could have impacts on fisheries, We'd have to watch out for, boater, there's lots of complications. But it's certainly something that's been kicked around. Good question.
Careful what you wish for. I have a question that was emailed to us. Could human sewage be a factor in the Lake Superior blooms? Large rainfall also causes storm drain issues, causing the release of human sewage.
Yeah, so human sewage, I mentioned historically human sewage was the main cause of these harmful algal blooms. And this was really before we treated sewage, right? Before it went into natural waters. And human sewage certainly in many smaller lakes, we didn't talk about small lakes today, we focused on the Great Lakes, but in many smaller lakes, like go up north, septic systems and leaking septic systems can certainly be a major source of nutrients to lakes. On the scale of Lake Superior, we don't think that that's a major source of nutrients, and it's just a matter of volumes and nutrient budgets. So we think that most of that, sewage could certainly contribute, but we think that most of it is coming off the land surface as opposed from human sewage.
Hi.
Hi.
The good thing about HABs is that there's a visual indicator, like as long as you see bright green water.
You can see it, yes.
No way to keep yourself safe. But you talked about one of the invisible effects, which is like the aerosol toxins.
Right.
Are there other invisible dangers of HABs? Like, do the toxins stay generally in the algal mass, or do they drift at all into other water?
Yeah, that's a great question. And if I had brought a tube of the toxin over dissolved in water, you can't see it, the toxin itself, and the toxins can be released from the cells, so they can be separated, and so it's theoretically possible that you could have toxin in water that you can't see, but in practice, usually the toxin is directly associated with these scums. So luckily, that visual cue of green is usually a really good indicator of high toxin levels. I'm not aware of any examples, in natural lakes at least, where you have clear water and high toxins. I think where it could become an issue, there have been some examples, like the biggest mass human mortality event was where water contamination from the field got into a medical environment where they're using that water for dialysis in Brazil, and a number of people were killed because of that. So there's some kind of niche examples of how that could happen, but generally in a lake, if the water's clear, you should be okay, at least from the perspective of algal toxins.
Okay, that brought up a follow-up question.
Yeah.
Can drinking water plants treat algal blooms?
They can. Yeah, there are methods of treating it, but they're expensive. So they use things like ozone to kill the cells and things like charcoal to bind and remove the toxins, but they're expensive. And the city of Toledo, for example, has invested hundreds of millions of dollars in water treatment that can deal with these toxins. So it's another example of the costs of these harmful algal bloom events, right? We're paying for our cheap food with more expensive drinking water treatment and more expensive incentivization of farmers to use best management practices. So it comes back to get us.
There's a couple of related questions I'm going to try to lump together. One is about mutations from benign to harmful and vice versa.
Yeah.
And just adding to that, are there in fact benefits, for example, CO2 uptake and so on, that you can put into the big picture here?
Right, good questions. And maybe some research avenues here. So we are getting to some good questions, science questions. So the first question, so actually let me address the benefits. So these organisms are certainly taking CO2 out of the atmosphere and putting it into their biomass. And if those organisms were to sink to the bottom of the lake and stay there, that could be carbon sequestration. But typically the organisms are broken down and degraded pretty quickly. And so that CO2 is released right back into the atmosphere. And the other issue is that decomposition is also often associated, we think, with production of other greenhouse gases like methane and nitrous oxide. So actually, if anything, these harmful algal blooms are probably contributing greenhouse gases to the system, okay? So not necessarily beneficial. The main benefit that we're looking at is we're thinking about these harmful algal blooms as potential treasure troves for medicinal purposes, discovery of new antibiotics and new anti-cancer compounds. So we have some discoveries there that we're excited about. And the other part of the question, remind me, Tim.
About mutations?
Mutations, yeah, mutations are super interesting. The genes that produce the toxins can't just, they can mutate, but they can also actually be transferred between organisms. One of the great powers of bacteria is that they can share genes. So not only, not only can I pass my genes to my offspring, my kids, I can also share genes with people in my immediate environment. It's an amazing power of bacteria to do so. So they can actually spread these genes between organisms. Our data indicates that type of mutation is most prominent on timescales of hundreds or thousands of years. So it's certainly occurring on short timescales, but it probably doesn't affect bloom dynamics like on seasonal timescales. There's one more dimension to that I want to mention about mutations, is that these harmful algal blooms are also thought to be reservoirs and vectors of antimicrobial resistance. So antibiotic resistance. So there are bacteria that are associated with these harmful algal blooms that carry antibiotic resistance, which is a big public health concern. And so I have a PhD student that's studying that, is how are harmful algal blooms potentially involved in spreading antibiotic resistance. So mutation and evolution is certainly key to the health impacts of these harmful algal blooms.
Wow.
You had a slide that mentioned this concept of genome binning and that this was something that's cheaper and easier nowadays. Can you explain a little bit about that? What is this, a machine learning type thing, or?
Yeah, sure.
And maybe one other thing, you mentioned 10 years ago that you had an inspiring question. Could you share what it was?
Sure. Okay, so first of all, yeah, thanks for the question about genomics. This is something that my lab focuses on that I'm really excited about. So first of all, just to give you a sense, that this DNA sequencing revolution has really taken off in the past 20 years or so, right? So in the early 2000s, it cost about $1 billion to sequence a human genome, and now it costs about $1,000 to sequence a human genome. So it's one of these technologies, one of the few that has outpaced advances in computer technology, for example. So this is faster than Moore's law of how fast computer processors increase. So it's really amazing technological advancement. And the question you asked about binning is a perceptive question. So oftentimes when we get these big pieces of DNA, we don't know what organism they belong to. And so we bin them, we assign them to different genomes based on signatures that are inherent in the DNA, not just the sequence, but patterns like, we call them tetranucleotides. So the different four-letter words and their frequency, right? So there's these weird patterns that are associated with certain species, and we do use machine learning to recognize those signatures and to place the sequences into the right genome. So there is a lot of machine learning involved there. So this is really computational science. So I have a full-time computer scientist that helps run my laboratory. Your other question was about, what was the interesting question last time? Well, last time I talked about how these cyanobacteria oxygenated the Earth's atmosphere. And I talked about how under low oxygen conditions, the amount of time that organisms have is really critical to how much oxygen they produce. And a question from the audience asked, so what about when Earth's day length was shorter? So how many people knew that Earth's spinning, this is good with daylight savings coming on, Earth's spin rate has been slowing down over time. So on the early Earth, it would spin around every 6 to 7 hours, it's estimated. We now have 24-hour days, we had 6 to 7 hour days. So his question was, and I didn't even know about that when I gave the talk. So his question was, okay, so if day length is important in terms of how much oxygen is produced, what effect did changing day length have on oxygen production? And so that led to a collaboration where we asked that very question and found that Earth's change in spin rate through time likely had a tremendous impact on the amount of oxygen produced, and therefore the timing of Earth's oxygenation and the rise of life as we know it.
Wow.
So it's a great example of how planetary things affect biological things. So amazing that we're all here based on this confluence of events, right?
Here's a complex question. There are a couple actually, but is there a bacterial phage that kills this cyanobacteria, and could it be used to control the bacteria?
Yes, good question, Tim. This is a good idea for another speaker. So Melissa Duhaime is a professor in ecology and evolutionary biology who is studying the viruses that infect the cyanobacteria. And so this is thought to be the major cause of mortality of cyanobacteria. And unfortunately, the cyanobacteria have their own little microbial immune systems, and so as the viruses come and infect the bacteria, they develop immunity to those viruses, and there's this arms race called Kill the Winner, where the most abundant strains of cyanobacteria are being killed, but then they develop resistance, and then new viruses come. So there's this arms race between the viruses and the bacteria that plays out in Lake Erie, and Melissa is trying to study that, to understand it, but also perhaps to develop solutions to targeting these cyanobacteria.
Mm-hmm.
Yep.
And you also just briefly mentioned this, but maybe you could say a little bit more about and there's actually a specific reference to Wang et al., but about the antibiotic-resistant gene reservoir in the cyanobacteria.
And I think this is from a listener who emailed me yesterday. I haven't had a chance to get back to him, but there's a paper, I think from Lake Taihu in China, showing that these microcystis blooms can be a reservoir of antibiotics, right? And it hasn't really been studied in Lake Erie. But as I mentioned, I have a PhD student that is delving into these genomic data sets to ask that question. Are antibiotic resistance genes associated with these harmful algal blooms? And if so, is that coming from the agriculture or from the human wastewater treatment? You can actually measure, this is amazing, go out offshore, for those that are boaters, go out offshore into the middle of the Great Lakes, you can measure antibiotics in the water. You can measure caffeine in the water. You can measure all types of pharmaceuticals in the water, acetaminophen, right? So an example of how we're affecting the chemistry of our natural waters. And so the question is, are these organisms coming from the antibiotic resistance? Is it coming from agriculture? We use a lot of antibiotics in agriculture. Or is it coming from humans that go, the antibiotics pass through our bodies, go to the restroom, goes through the sewage systems, and ends up in the lakes? And how is that all affecting the microbiology of the lakes and the spread of antibiotic resistance genes? So that's a question I think that's open right now for the Great Lakes, but we're starting to study it.
Sorry, this is not a science question. I'm an English major. I was wondering, going back to your diagram about the decision support tools,
Yeah.
if anyone on your team is kind of going back and looking at the effectiveness of various approaches and how that might inform policy for agricultural production, like on any scale.
Yeah, that's a great question. And my team at Sigler is mainly focused on what's happening in the lake. And this is kind of an artifact of how U.S. federal agencies work. NOAA works in the lake, and then it's typically the U.S. Geological Survey and then the various agricultural agencies that work on the land. So we're not as involved in the agricultural practices, but there are people in the room. Allison Bressler is here, who works at the University of Michigan Water Center and interacts a lot with those agricultural agencies. And they are very much looking at the effectiveness of the different practices. So we have both in the state of Michigan and the state of Ohio, a lot of research going on at Michigan State and Ohio State that's looking at the effectiveness of these best management practices, absolutely. And my view is that there are still scientific questions there, but we kind of know what the best management practices are. The big challenge is getting farmers to do it. And there's a lot of, because we can't mandate it because of the environmental laws that we have, I think that the main way forward there is social science, understanding behaviorally what is it going to take to get these farmers to participate in these programs. And what's coming out of Ohio, I think, is some good evidence that strengthening networks among farmers and cooperation. And once you get one farmer finding, like, not only do these best management practices reduce nutrients, they also improve the productivity of my farm. That's when you really make inroads and you have these networks of collaboration and communication. If we need to infiltrate those networks and spread the word, right, about these best management practices, I think is the real opportunity. Yep.
The couple of questions related to the temperature dependence of the growth rate. Mm-hmm. That's actually the growth rate, not the actual population. One of them is, there was a peak in the cyanobacterial around 35, 37 degrees. That's like really hot, right?
Yeah.
So we're not going to get close to that where it turns over. But the related question, I just wanted to clarify that, but the related question was, is there a reason, like bacterial competition, that the saxotoxin is being produced at higher temperatures?
Yeah. So
yes, you throw the three different species, right?
Yeah. So the observation is that the cyanobacterial maximum growth rate occurs in the 30s, right? So 30, 31, 32. Maybe someone can Google that. What is that in Fahrenheit? It's, I think, in the 80s Fahrenheit. So typically,
90-ish, yeah.
low 90s even. Yeah. So 37 we know is our body temperature, 98.6. So we don't expect Lake Erie to get that warm anytime soon. But we are seeing temperatures in the upper 20s in the summer. And if we're changing a degree per decade, right, it doesn't have to be too many decades before we get there. But the observation is correct that, this is sort of on the upper end and we're not going to get there, which is hopefully good news. But the other thing I would say is that this is really oversimplified. Cyanobacteria is a whole phylum of organisms, and there are many different genera and species within that, and so many different cyanobacteria have different growth optima. It's just that in general, they have higher temperature optima compared to their competitive algae, okay? But the observation, I think, is a good one, Tim, that we hope not to get up to the 30s. This can be an issue, though, in more tropical places or subtropical places. Like Florida has some major harmful algal bloom issues. Lake Okeechobee in the central part of Florida has major issues with microcystis, and some of the microcystis strains down there grow at these higher temperatures, and down there you can talk about getting into the 30s for lakes. So probably not, hopefully not in the Great Lakes, but there are other locations where that's possible.
I have to comment, this I think is the largest number of online questions that I have seen since we started doing this.
Does that
So
mean
this
I
is-
was
This is an
unclear, or is
amazing
it-
topic. But we also have people here, so please.
I was just wondering, how much effect does human activity like boating have on algal growth?
Yeah, good question. I don't think it has a big impact on the algal growth. These lakes are just so big that the human activity, I think, is kind of a drop in the bucket. What it can affect, though, is, especially locally, for anybody that's been on a speedboat, for example, like there's a wake behind that speedboat, right? And so that's a major potential source of these aerosols. So if you're out there on a jet ski, I mean, I think everybody has a sense of how much water a jet ski can kick up. That is certainly a way that you can kick toxins up into the air. So I think on a boat, you're probably killing some cyanobacteria as you speed through, but you're not putting a dent in their populations. I think the bigger concern would be with the aerosolization of toxin.
Oh, no, I was reading the other way around because I read online that the pollution from boating can actually help them grow.
Pollution from boating can help them grow. I'm not familiar with any of that. I would think, especially maybe in small lakes, but I think especially in big lakes, that the amount of pollution you're getting from the boats themselves is probably not enough to make a dent in the nutrient budgets, is my guess. Yeah, please send it to me. Hadn't heard of that.
From online, there are two related things. One which I'm going to truncate significantly, but is the Conservation Reserve Program in Ohio, H2 Ohio, under threat, do you think? And also, how helpful is Canada? So those are two sides of Lake Erie.
Yeah,
Right.
our neighbors, yeah. I'm not as familiar with the H2 Ohio program. Another good suggestion here would be Dr. Sylvia Newell, who joined the University of Michigan as Director of Michigan Sea Grant. She came from Ohio, where she worked pretty closely with H2 Ohio, so she's very familiar with it. But what I do know is that program has to be renewed every 2 years. And I think the funding level is up to something like $150 or $200 million, and it has to be renewed every 2 years, which does make it in jeopardy due to the political winds of that particular time period. But I think overall my understanding is it's got pretty widespread support because people are so concerned with what's going on in Lake Erie. I mean, Ohio really relies on Lake Erie for a lot of tourism and economic activity.
Mm-hmm.
Yep.
In Canada?
Excuse me. Canada has been a good partner of the United States in Great Lakes issues. It's a really interesting example of water governance because we have two nations that the Great Lakes straddle these two nations. And so they have both signed on to this Great Lakes Water Quality Agreement. Both the US and Canada have pledged to reduce phosphorus levels by 40%. And so I think they've been a good partner. And in fact, we have substantial research collaborations as well. So Sigler actually has a Canadian partner, the Great Lakes Institute for Environmental Research at the University of Windsor. So there's really close collaboration and partnership from the research level all the way up to the policy level.
Mm-hmm. We'll take just a couple more, I think 3 more questions actually. Okay, go ahead, please.
Yes.
You mentioned Australia towards the beginning of the lecture.
Yeah.
When I went, I saw like a body of water that had the blooms, and it was like a very slimy texture. I got dared to touch it, and I love that. But you said that blooms like warmer ones, but Australia is more dry heat, and Michigan is more humid heat. So does that have any effect on like the way it feels, or?
Yeah, good question. What's going to matter most is actually the water temperature, not the air temperature. And because these guys are in close contact with the water, they're also not going to care as much about the humidity, that dry heat or warm heat. So it's amazing to me that in otherwise seemingly very different environments, like Australia and Michigan, you can have the same species of cyanobacterium, Microcystis aeruginosa. So again, it's mostly, I would say, about the water temperature and the amount of nutrients rather than the other meteorological conditions. Yep, good question.
There's a couple of questions about, I don't know what lake flips are, but it may be related, the idea of mixing cold and surface, so the lower levels of water temperature with the surface temperature.
Yeah.
Another engineering feat.
Yeah, yeah. That's also another impact of climate change. So what the question is referring to is lakes tend to thermally stratify. For anybody who's gone swimming, right, you felt that as you go down, sometimes you can go from the warmer water to the colder water down below. And that's because warmer water expands slightly and is less dense. And so once you have warmer water above colder water, that water will tend to not mix. And that can actually favor these cyanobacteria, because these cyanobacteria, one of their superpowers is they have little gas vesicles that they can use like a submarine to regulate their buoyancy. So they can travel up and down in the water column and go down for nutrients and come up for sunlight. So as the waters thermally stratify, that is going to favor these cyanobacteria. The question may be about, can we deal with these blooms by mixing the water? And that is certainly a potential strategy in smaller water bodies, like your neighborhood pond. But it would, I think, just take too much energy to try and physically mix, for example, Lake Erie, for that to be a part of the solution. Likewise, we have good chemical treatments for these cyanobacterial blooms. Hydrogen peroxide, which you put on your cuts maybe, is a terrific killer of cyanobacteria, and it doesn't harm a lot of the other organisms. There's just not enough hydrogen peroxide in the world to add to Lake Erie to kill these cyanobacteria. So it's really the scale of the problem in the Great Lakes that's the challenge.
And our last question here.
Does everything you talked about in your presentation apply to red tide on the southern coast as well?
Yeah. Great question. So red tide, you refer to the southern coast like it's in Florida, for example. Red tide is caused by a eukaryotic algae, so different type of algae, not cyanobacteria, but they do produce toxins. They produce domoic acid, and they can also produce saxitoxin. So it can certainly be a big concern. Red tide, actually, the science on aerosolization of toxins is much farther along in red tides, and we know that red tide domoic acid, for example, can get up into the air and cause respiratory issues. So I would say a lot of what we talked about here today applies to red tides, certainly nutrients and some of the temperature effects, but because they are different organisms, not everything applies. Yeah, good question.
Well, thanks everybody for your great questions.
Yeah.
And also online, and we'll make sure that you get to know about questions that we didn't get to ask you here. And thank you so much, Professor Gregory Dick, for that amazing presentation.